Hello, folks. Here we are. It is Wednesday, January 20th. And today what I want to do is I want to go over exactly how our genetic engineering works. That's the goal of this week, is so that you understand this. This technology is revolutionary. And when I say revolutionary, I don't think any of us really grasp how much it will change our world in the next 10 to 15 years even. We are now understanding how this works so well that the alterations that we're going to see in different species and including in people is going to be mind-boggling. In the next decade, diseases that everybody's needed to deal with may become a thing of the past. Wow, my hair is going nuts up top. Wow, look, I have a little bit of hair. Look, you can see it. How cool is that? All right, anyway, let me talk about how this works. So at this point, ignore the organism. The organism is not going to be important towards what we're doing right now. All right, but if we take a look at the whiteboard here, you can see we have this gene in black. This is the host. So this is the organism that we want to modify. All right, and once again, it's A's, T's, C's, and G's. We could write down the other side of the DNA chain because we know that A links to T, C links to G, and then vice versa. T links to A, and G links to C. Always. We know that, but because we know that, we really don't need it. Instead, we just look at one half of it. And when we do, you see this long sequence, and this can be thousands and thousands of letters long. All right, but we want to include a new trait to this organism. Now, this new trait, you can already see I have written up here. All right, so this gene up top, let's imagine that this is, I don't know, let's say that this makes a protein, like an antibody. All right, let's just go with a protein. All right, it makes a specific protein. And let's just say that it makes some membrane protein, right? A protein that's embedded in the cell membrane. And this is what we want to become a part of this. How do we do that? You can't just take it and force it in. It doesn't work quite that easily. What needs to happen is we need to find a section of the host that we could remove and then replace with this one so that this gene now gets read instead of the host gene that it no longer needs. Now, why is this important? Why do we need to do it that way? Because your DNA is full of all sorts of spaces that don't do anything. Huge sections that we don't yet fully understand. Do they have a purpose, or are they just garbage DNA? Don't understand. A bunch of our DNA is from viruses that have written themselves into our DNA over the millennia that our species has been evolving. And so when we look at it, if you were to put this anywhere and you just shove it in, you might put it into a section that's no longer used, no longer read, and no longer has function. Or you could put it into a section that's all about those switches that turn things on or off, and we don't want to mess with those. So instead, what we have to do is we have to find another section that codes for another protein, and we'll cut that part out, and then we'll plug this one in. Well, how do we do that? What we do is we identify these letters before the gene that we want and after the gene that we want. Now, by identifying very particular setups like this, we know that those are areas that get used. So now we have to check our host, and we have to identify, do we see these same things? We do. Right here, GCA shows up right here. ATC shows up right there. So now what we will do is we'll use an enzyme. Our enzyme will come in, nom, 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 like scissors, like Pac-Man, and it will nibble away this portion. It'll cut it here, and it'll cut it here. And once it does, this middle piece floats off because it's now been severed, right? That is no longer a portion of this genetic code. Then we just need to add our goal. Add this into the mix. It will automatically connect and line itself up in that gap because it's looking for a G, a C, and an A, and an A, T, C on each of those sides. And then the rest of this just comes along with it. So now we get our C, our A. I didn't think about spacing. Right? So now we have this new membrane protein into this host DNA. We've removed one protein that it makes, and now we have given it a new protein to make. This is the essence of genetic engineering. And while it may seem really complex, this process, there go the lights, hold on a moment, but this process is getting easier and easier, literally every day. In the last few years, literally just the last 3 or 4 years, we have seen the cost of genetic engineering plummet by almost like 90% as new technologies become available. Now, there are some limitations to this because thus far, we can't do big changes. What do I mean by big changes? We cannot do something like taking a kitty cat that's my kitty cat. We can't give it wings. All right. Why? Well, because in order to give a cat wings, we're not changing a protein. We have to change the entire structure of the organism, and we do not have a good handle on how to do that yet. First of all, if we gave a cat wings, it doesn't have front legs anymore. There is no mammal, reptile, bird. There is no chordate, nothing with a backbone that has wings and legs. Only insects, and they follow very different rules. So when we look at a bird, it's their arms that have turned into the wings. It is their shoulders that give them the musculature in order to fly. So this cat would need to change wildly. The wings alone, even if we could do this, wouldn't be enough to let that cat fly because now we would need to shrink its bones so that it's lighter. Well, now that means that they can't have the amount of muscle that they do because that could like fracture or break something. So now we need to change muscle attachments all over the body. It gets very complex. Right now, all of our genetic engineering is all about taking a cell and teaching it how to make something new, right? A new protein, a new something. I'm going to see if the glare is just in the way. All right, but that is the goal right now, is to get them to make something new. And sometimes that's amazing. Sometimes that's all we need to completely cure some sort of genetic disease or condition that somebody is suffering from. Sometimes it is as simple as a single change. Case in point, there are a number of diseases out there where if we could go into their genetic code and we could just change one letter, they have one letter that's wrong. So instead of an A here, they have a T. And that alone causes the entirety of their problem. There it is. If we could come in with our good gene, the healthy, normal one that produces the right thing, and have that replace this bad one, correcting that one letter, all of a sudden, their health would improve. They would be fine. The glory, the promise, the potential for this is astronomical. Imagine a world where nobody suffers from things like cystic fibrosis, muscular dystrophy, where all sorts of other genetic conditions, color blindness, hemophilia, could all be cured by a simple injection that introduces the good copy of that gene with the right tags on either side to get it there. And then your cells, the machinery in there, does all the rest of the work. And it takes out the bad, puts in the new. That is what genetic engineering right now offers us. Not just better foods that grow faster and more conditions, but this is the goal process right now to really just improve human health and that of other organisms. So, ladies and gentlemen, I hope that that helps explain the reality currently of how this technology works. As we move on with it, as we get better, we will encounter and we will master new versions. We will understand the process better. And one day we might be able to make that flying cat. But not today. Probably not tomorrow. Maybe not in our lifetimes, but one never knows. Ladies and gentlemen, your only task today is I want you to think about what would you like to see changed if you had a lab and the materials and the people necessary to genetically engineer an organism. What would you like to do? Once again, try to keep it reasonable. We're talking about altering a single thing that's made, not a complete organism. And the trait that you want to include has to come from something that already exists. We can't genetically engineer something to breathe fire unless we can find something else and identify the genes that allow it to breathe fire. So, ladies and gentlemen, that is our task. And as always, I can't wait to see you in class.